All right, good morning, everybody. Hello, hello. Give me a quick yes in the chat if you can hear me. Awesome, great. All right. So today is a very exciting day because we are going to talk about cells. And let me kind of frame this, and hopefully this is not super surprising to anyone, but a lot of the stuff that we'll be talking about is certainly covers some background that you should already have from cell biology, but we're spending just this one chapter on a lot of the cell structure function stuff that you might have spent more time on in cell bio. And while we certainly will go over the eukaryotic stuff again that's important for microbes, we're going to focus more on maybe prokaryotic biology than you have in the past. And also spend some time kind of comparing these two cell types to each other and really pointing out some of the key features in prokaryotes and eukaryotes, but mostly prokaryotes, that are clinically important. So here's kind of the big picture where we're going with this. We have to talk about what cells have in common. So what are features that all cells have? Like when we say something is a cell, what do we know for sure about it? We will talk many times, but we're going to have a specific point talking about the differences between the two major cell types, prokaryotes and eukaryotes. And then we'll go through the plasma membrane, the cell wall, and various cell structures that are found in prokaryotes. And on Thursday, we'll talk more in depth about the eukaryotic organelles and structures that are important in this field. So I just want you to take a second to think about what you know. Write down or type out the structures that all cells have just for yourself in a second. You don't have to put anything into the chat. If you're just joining us, remember to sign in by putting your last name, first name. And just think about it in your mind and write down on maybe on a piece of paper what structures you're very confident that all cells have. And then once you have that list, think about what the major function and composition of those structures is. If you say, well, all cells have a blah, what's that blah made of? And what does it do? What is its major role? I'll give you one more minute to kind of come up with the list that you're very sure all cells have, and then think about what they're made of and how they work. If you're just joining us a few minutes late, don't forget to sign in, last name, first name. Okay, so with that kind of preparation, that mental preparation, we're going to go through. Now what I want you to do is go get your participations box, you know, that one that has the green check mark and the red X, and also where you would raise your hand and stuff like that. So go get that participations box out. And as I put up a structure, you're going to say, yes, this is something all cells have, or no, only some cells have it, right? So it could be a structure that some cells have, but if not every cell has it, you have to say no. If you're certain all cells have this, then you would say yes. So go ahead and vote. Green check mark for yes, all cells have a cell wall. Red X for no, they don't. Excellent. Okay. So most of you put no, and that is correct. So many cells have cell walls, right? Importantly, but not all of them do. And for instance, you, and I'm sure that you are the most important creature on the earth to yourself, do not have any cell walls. Cell membrane. So I'm just going to clear that and go ahead and vote again. Cell membrane, yes, or cell membrane, no. Are there cells that don't have a cell membrane, or do all cells have a cell membrane? Great. The answer is yes. That's one of the defining features of cells. They have this phospholipid bilayer boundary around them. What about cytoplasm? Let me just clear that box and go ahead and vote again. Cytoplasm, yes, or cytoplasm, no? Great. Most of you put yes, and that's accurate because that's the fluid-filled interior of both prokaryotic and eukaryotic cells. Now, what about a cytoskeleton? Do all cells have a cytoskeleton? Go ahead and vote. Great, most of you put no, and that's correct. Eukaryotic cells have a cytoskeleton. Remember, it's got those 3 different filaments, the actin, the microtubules, the intermediate filaments, and we'll talk more about those on Thursday. But prokaryotic cells do not have a cytoskeleton. So the functions that are fulfilled by the cytoskeleton in eukaryotic cells are provided by different structures in prokaryotes. Okay, this should be an easy one. All cells have a nucleus or all cells do not have a nucleus. Please vote now. Excellent. Okay, so you were alive in cell biology because you learned that eukaryotic cells are defined by the presence of a nucleus and prokaryotic cells are defined by the absence of that nucleus. Okay, now what about DNA? All cells, yes, have it, or all cells, no, do not have it. Or I should say some cells have it and some don't. Great, yes. All cells have DNA. That's the instructions for making all their proteins. What about ribosomes? Do all cells have ribosomes? Good, yes, they do. Okay. What about lysosomes? Lysosomes. Oh, I already gave the answer away. Sorry, that was my bad with a little mouse click, but most of you were putting no anyway, so you already knew where I was going with this. Lysosomes are an organelle, and like all organelles, they're restricted to eukaryotic cells. Good. Okay, so now what we're gonna do is a raise your hand. So I asked you at the beginning to think about like what structures cells have in common, what they're made of and what they do. We're gonna go through some of those structures. Not all of the ones that I'll mention, most of them are common to all cells. There's one that I'll mention that's not, it's only in some cells, but still I want you to think about or answer. So for this cell membrane, if you can identify its function and or its composition, raise your hand. Go ahead, Denise, tell us what you know about cell membranes.
They exchange nutrients, waste, and information with the environment.
Yes, so they do exchange. Excellent. I'm gonna that was a fantastic answer. I'm gonna poke Luke to tell us what he remembers about their composition. Go ahead, Luke.
They're polar and they have a phospholipid bilayer.
They have a phospholipid bilayer, absolutely right. And the outside and the inside of the membrane, so the 2 sides are polar, the interior is a non-polar sort of fatty acid mixture. Good, all right, so phospholipid bilayer has this job of exchange, and also it's got proteins in it, right, that are what do the exchange. So we would say exchange is its primary function. Also, interaction, and you might think, well, we're in microbiology and mostly talking about single-celled organisms, but even, and so when we talk about like human cells or multi-celled creatures, definitely the cells are interacting with each other at the cell membrane level, whereas microbes you might think are more solo, but they still have to interact with their environment and with each other. So for instance, some single-celled eukaryotic yeasts interact by finding mating partners, so they'll send out chemical signals, and so that's a spot where that happens as well. So exchange of nutrients and waste and information, and also interaction with other cells in the environment. And what are they made of? Phospholipid bilayer, as we heard, and also proteins that do the job of exchange. Okay, now cell walls are not found in every cell, but in those cells that have it, what is the function and or composition of the cell wall? Evelyn, go ahead. What would you like to share?
For the function of them, it's to protect the cell and to make sure like the cell remains its shape.
Yeah, so protection and shape. Cambria, you also had your hand up. Would you like to add anything about the function or composition of the cell wall?
Doesn't it specifically protect it from lysis?
Yes, good. Excellent, good answers. Okay, so protection, cell wall offers protection from lysis, that is osmolysis or explosion due to having too much water come inside. So often cells are found in a world, especially microbes, not so much your cells because we're in this homeostatic environment that's very carefully regulated, but most microbes will find themselves in a world where the osmotic pressure is unpredictable. and frequently there's more water outside than inside. The water wants to come in, and so the cell wall is a way to protect the cell from exploding if that happens. It also, and especially in prokaryotes, helps determine the shape of the cell. And this is more commonly conferred by the cytoskeleton in eukaryotic cells. The composition depends, okay? So nobody really ventured into that, and it does vary quite a bit depending on the cell type. And in fact, the composition of the cell wall can be a diagnostic that is helpful in determining what we're dealing with. So there's always carbohydrate component to the cell wall. It's heavy in carbs, but depending on whether you're bacterium or an algae or a fungus, then there may be other components. Okay, let's see what we know about the function or composition of the ribosome. Stacy, go ahead.
Its function is for protein synthesis, and it's made up of RNA and protein.
Good job. Yes, absolutely right. So its function is protein synthesis, and it's got, you want to imagine it like a big machine, okay? Even like something crazy like a bulldozer, right? So it's got pieces. It's got the wheel, and it has a bucket, and it's got gear shifts and windows, and a metal body. All those different pieces are made of either RNA or protein. And so if we go to my silly analogy of this, of this bulldozer, if you think of a bulldozer being sort of yellow and black, you could imagine some parts are made out of our RNA, and that's the yellow part, and some parts are made out of black, are black, and that's the protein part. So when we get to genetics, I want you to try to remember that protein has a three-dimensional shape and can assume a function as a result. So it could be like, the wheel or something, but RNA also can do this. All right, finally we come to DNA. Trin, what would you like to tell us about the function and or composition of the DNA?
DNA functions to give instruction to make proteins.
That's exactly right. Yep.
It's a double-strand nucleic acid in like a helix shape.
That's correct. Okay, so its function is to provide the information for how to make proteins, or the recipe for how to make the proteins that are used in the cell to do everything. And well, except for RNA also does some things. And its composition is a nucleic acid, deoxyribonucleic acid, and it's two-stranded, like that double-stranded helix that you guys remember from genetics. Great, okay. So I just saw a lot of hands go up, and I want to make sure if you had a question, and not just wanting to answer my prompts, but if you had an actual question, go ahead and raise your hand just so I don't miss something that you were curious about. Let's take a second to. Okay. All right. So you'll remember from cell biology that there's two cell types, prokaryotic cells and eukaryotic cells. And one of the major defining features is whether or not there's a nucleus. Although that's not the only thing, right? They differ in terms of their size. The prokaryotes are usually a bit smaller. They differ in terms of their contents. Eukaryotes have organelles, prokaryotes don't, and in a variety of other ways. So let's see, if we just focus on, do you remember from the first week, we talked about all the different types of microbes that we would cover in this course. So we're going to focus on the cellular ones right now. Obviously things like viroids and prions and we're not going to worry about, and viruses we're not going to worry about right now, but we're going to focus on cells that are microbiological. And I'm going to ask you if they're prokaryotes. So if they are prokaryotes, you'd pick that yes check mark, the green check mark. And if you think the cell that I'm mentioning is not a prokaryote, it's a eukaryote, you would say no, that's a eukaryote, okay? So bacteria. Use your participation box, click the green check mark for yes, it's a prokaryote, or X for no, it's a eukaryote. Great. Okay, so most of us know, thankfully, that bacteria are in fact prokaryotes. What about an amoeba, which is part of the protozoans? An amoeba that is protozoan. Excellent. So most of you said no, that's correct. Amoeba, or any protozoan, amoeba is just one example, are these animal-like single-celled eukaryotes in the kingdom Protista. Okay, algae are also microscopic. Algae, are they prokaryotes, green check mark, or eukaryotes, red X? Good. Little bit of a mixed answer there, but for the most part, people picked no, and that's correct. Algae are plant-like members of the kingdom Protista, and so they are microscopic, but they are eukaryotes. All right, so let's go to our yeast. These are single-celled fungi. Are they prokaryotes, green check mark, or eukaryotes, red X? Good, yes. Fungi are all eukaryotes. Some, like yeast, are single-celled and microscopic, and some are large and, you know, you can see them growing in the forest. Okay, domain archaea. Are they prokaryotes, green check mark, or are they eukaryotes, red X? Good job, everyone. Good. So, yes, archaeans, like bacteria, domain bacteria and domain archaea, are both prokaryotic cells. That means they're smaller, less complicated, and they have certain distinctive features. So this is straight out of your video lectures that you watched already. And so this is sort of defining the features of these prokaryotic domains, the archaea and the bacteria. They're small, okay? So, you know, it can be a little confusing because the size range is very broad. You know, this is an average size range. If you read 2 textbooks, they're going to give you different ranges. So some will say like 0.2 to 3 and 0.5 to 5, and I say around 1 micrometer. But they're all around this range, like this, like really low numbers of micrometers or even fractions of micrometers, okay? So, and these smaller numbers like 0.2, 0.3, 0.5 are usually for rods, and they are the width. And this other measurement is usually the length of rods, and this would be like diameter. Okay, so in comparison, eukaryotic cells can range from 10 to 100. Now, in fact, there are examples outside this range. So yeasts, like budding yeast, is a little bit smaller. It's actually like 6 or 7. And there are algae that are like 400 or 500, and protozoans that are also pretty large. But this is sort of an average for human cells. This is like the smallest human cell and sort of like a larger one. So on average, eukaryotes are between 10 and 100 times larger than prokaryotes. Eukaryotes have a true nucleus. They typically have multiple chromosomes. Prokaryotes only have 1, and it's circular. Eukaryotes have organelles, prokaryotes don't. If there's a cell wall, its composition is different. So you can actually, if you see a cell and it's microscopic and it has a cell wall, just by looking at the chemical composition of the cell wall, you can assign its what category it's in. Okay, so bacteria have peptidoglycan cell walls, algae have cellulose, and fungi like yeast have chitin, and archaeans have something else entirely. How they reproduce is different. Much more complicated process of mitosis in eukaryotes, simpler process of binary fission in prokaryotes, although there are other examples we do not cover of ways, you know, things like segmentation and things like that, budding. Ribosomes present in both cell types, but they're a little bit different in their structure. The eukaryotes, one way to say it is that they're bigger, and that's true, they're slightly larger in size, but more importantly, more dense. They can, not all cells have flagella, but those that do, the prokaryotic flagellum is made of this protein, flagellin, and the eukaryotic flagella is made of something called microtubules, and it's actually larger and it moves differently. And we'll cover that in more detail later on. And finally, even though all cells have a cell membrane and their composition is actually pretty similar, the function of the cell membrane differs a little bit. There's extra stuff that the eukaryotic membrane can do that the prokaryotic membrane cannot. Okay, so let's just take a break from all that information and ask a question. So in 1932, penicillin was discovered, and it was called a miracle drug. And the reason for that is up until that point, treatments for disease, especially infectious disease, typically had a lot of side effects. And that was because they weren't very specific. Whereas penicillin specifically impacted some infectious diseases and didn't harm people. There really weren't very many side effects, you know, unless you have a penicillin allergy. It's a pretty safe thing to take. What's the reason for that? Raise your hand if you know. What's the reason for that? Kylie, go ahead. Tell us what's the reason.
Because it damaged the peptidoglycan walls for the bacteria.
That's right. So the target of this drug is a chemical component in some bacterial cell walls that is not present in humans. And so it's true that it targets peptidoglycan, although as we'll learn today or review today, it targets a part of peptidoglycan called the cross bridges that's only found in gram-positive. So it's even more specific. It doesn't kill all bacteria, just the gram-positive ones. Okay, So I'd like you to think about this for a second, and then I'm just going to cold call some people for some answers. In general, why is it important to understand the structure, the nitty-gritty details of prokaryotic cell structure? Why is that important? Just think about it. What your reasons might be. Like, since prokaryotes are different from us, why do we care about their structure? And then I'll cold call just a couple people to get your answers. Okay, let's go to Amanda. Amanda, what would you say in response to this prompt? Why prokaryotic cell structure matters?
Hello. Since they have a high surface area to low volume, that is beneficial to them. And then also their size allows them to do a rapid uptake of nutrients and excretion of wastes.
Yeah, so you're sort of touching on this really one of the important distinctions between them and us. And one of the implications of that, this one partly explains why they can grow faster. Excellent. Let's move on to Natalie Dixon, if you're able to unmute and say hi. Tell us what your response to this. Why does prokaryotic cell structure matter to us?
Maybe because to know if they're harmful or not.
Yeah, that's a great answer. Yes. Knowing about their structures can tell us a bit about whether they pose a threat to us or not. Absolutely. Let's go to one more person and let's ask, let's see, Erica. Erica, I have two Ericas actually, I'm so sorry. And you both have M as last name, Mucciaroni. I think I just butchered your last name and I'm so sorry. But if you're able to unmute, correct my pronunciation and then give us your answer, I'd love it.
Okay, Mucciaroni, can you say the prompt one more time? Sorry.
Yes, absolutely. So what would you say to this prompt? Why does the cell structure, knowing the cell structure of prokaryotic cells matter, given that they're different from us? Why do we care?
I would say the function of the cell, the shape.
Sure, the structure tells us a little bit about shape. Sometimes we will be able to see that under the microscope. Great answer. Let me just also, I see a bunch of hands. A lot of people got excited about sharing their ideas. And so I'm going to actually ask a few people. Haley, your hand was up. What would you like to add?
Can you hear me?
I can.
So I just wanted to add, in case, like, if we need to be prescribed antibiotics, it's important for whoever to know that, like, whatever structure they're targeting is not in our, like, eukaryotic cells, but it's just targeting the bacteria.
Oh yeah, that's a great answer too. Yeah, so understanding what the structure is of, say, something that might be causing an infection would allow us to pick a drug that would be useful and also not harmful to us. So knowing what their differences are means we can target drugs that are honing in on those differences. Good answer. Okay. Lauren, did you have anything you wanted to add? I see your hand went down. Okay. All right. Good answers, everyone. Thank you for sharing all those thoughts. Okay. So what we're going to do now is, I actually might postpone this a little bit only because I haven't finished creating my breakout room, so I might need some time. I'm going to postpone this to the break. And what I'm just going to give you sort of a heads up now. What you're going to do after we come back from the break when I've created the breakout rooms is you're going to do a little like group work with each other. And so you're going to look up what certain antibiotics do, like what their actual target is. So every antibiotic's kind of specific in what it's actually going to affect. So there's a bunch of them and you'll work together as a group to figure out what things do what. Okay, so we'll talk more about this on the break, after the break, when I've had a chance to create your breakout rooms. I'm only about halfway through doing that. Okay, so let's talk now about what you can see if you are looking at prokaryotic cells under a microscope, under a light microscope using a simple stain. Just write into the chat what things you'd be able to discern from that type of viewing or analysis. What can you learn about the prokaryotic cells if you just did a simple stain and looked at them under the microscope? Go ahead and write that into the chat. Good job. Okay. So pretty much, I see a lot of these in the chats. So you would be able to see the size. You can do a size estimate like you've been doing in lab. You can see their shape. That's the morphology. You can see how they're organized if they have a specific arrangement. Some of them don't. They're just random or single. And the average size, as we talked about for prokaryotes, is in this low micrometer range. It could be somewhere, like if it was a rod, it might be, the width varies. It could be as low as 0.2 micrometers up to 1 micrometer, and the length could be somewhere between 2 to 5, and a typical round cell would be maybe like, 1 or 2 micrometers wide. What else is there that's actually visible under the right circumstances that you can't see with a simple stain? Again, just focusing on prokaryotic cells only. Write into the chat something that could be there that we might be able to see under the right circumstances, but a simple stain is not going to reveal it. Great, so I'm seeing flagella, ribosomes. Cilia are a eukaryotic structure, so we're gonna set that aside for a conversation later. Endospores, the capsule. If I scroll back up, I thought I saw something else cool that you guys mentioned. And I'm sorry if I missed you. Okay. Whoopsies, I'm going, things are going crazy. Okay, so yes, all the internal structures that ribosomes you could see with an electron microscope or with a UV scope with, or a confocal under the right circumstances, not under a regular light microscope. Flagella, if we did a flagella stain, we could see capsules if we did a negative stain. And so there's other things that we can certainly view that are interesting to look at. Now, naming species is done by the people often that discover them. So it's not always a useful name, but sometimes it is. And the causative agent of strep throat is called Streptococcus pyogenes. The Streptococcus part is its genus name, and so it's grouped with a lot of other things that are also called Streptococcus, and here's its species name. What can you tell? What do you think you could at least have an educated guess about based on the name of this organism? What do you think you would be able to say about it with some degree of confidence based on its name? Put that into the chat for me. Great. Okay, so you're saying basically you'd be able to tell its shape is round because of the coccus part. That's true. And that it's got an arrangement that's chained. That's the strepto part. Good job. Okay, so its morphology is coccus or cocci, and it's arranged in chains, which is also known as strepto. Now, MRSA is something that if you're working in healthcare, you've probably heard of. It stands for methicillin resistant what? Write that into the chat if you know the answer. Methicillin resistant, what's the SA part? Good, yeah. Many of you have heard of the infamous Staphylococcus aureus. That's right, methicillin resistant Staphylococcus aureus. A really annoying creature because methicillin resistant is actually a step up from PRSA, penicillin resistant. So first the Staphylococcus aureus, because they're gram-positive, sensitive to penicillin, then they develop resistance. So we switched to using methicillin, then they develop resistance, and now we have MRSA. So then we switch to vancomycin, and now we have VRSA. Okay, but methicillin resistant Staphylococcus aureus, based on the name, what do you think you can say about it? What do you know about it? Just looking at the name, if you knew nothing else. Good. Yes, also a coccus arranged in clusters. That's the staphylode part. Okay, so now let's suppose someone comes into the clinic and they have an infection. And we look at the microbes, a swab of whatever, under the microscope. Does knowing the morphology and arrangement with a simple stain, would that be helpful in our diagnosis? Okay, yes, I agree. I think that would be helpful. Is it sufficient? If we look at something and we see clusters and they're cocci, can we say, oh, for sure, we know that's Staphylococcus aureus, and we know it's MRSA? Okay, good. Yeah, the answer would be no. We would have to do a little bit more work to be certain, because even though MRSA always looks like MRSA, there's also many other species that look like MRSA. And similarly, Streptococcus has this genus, which is sort of defined by its morphology and shape, has many species. So we don't know who we're dealing with necessarily. We could have a really strong idea based on symptomology and the person's lifestyle and their previous exposure or whatever, but it isn't the only thing that we need to be certain. Okay, and with that said, if we did a gram stain, on the other hand, we might have a little more certainty, at least about what antibiotic to choose. Okay, so let's look at, when we think about bacteria in particular, there's several layers that can be found, okay? These sort of external wrappings, if you will. And we talked about the cell membrane, we talked about its function and composition, its function in exchange and interaction, its composition of a phospholipid bilayer with proteins. What was the function, write this into the chat, of the cell wall? What was the function of the cell wall? Good. Protection from lysis and shape, absolutely. Mostly protection is its most important job, and it does also confer shape, which can be important too. It's composed of carbohydrates, and in bacteria, it's composed of carbs and protein. And some bacteria also have a lipid component. Now, what about the glycocalyx? What is its role? We haven't really talked about this yet today. Okay. So in terms of clinical relevance, yes, the glycocalyx can be part of attachment. We care most about it because in pathogens, the glycocalyx or capsule can provide protection from discovery. So your immune system will have a hard time finding encapsulated organisms, and it's made of carbohydrates. So do any prokaryotes, this question is, do prokaryotes have all three of these always? Great, yes, that's right. The answer is no, they don't always have them, but some of them do, okay? So those prokaryotes that do, what is the order? So I want you to go from the inside of the cell out. What is the order of the things that you would meet? If you're like inside the cytoplasm and you were working your way out, which things would you meet first? What order would you meet these different layers in a cell that had them all? Great. Yes, it's ABC. So we would, first we're in the cytoplasm, the first thing we're going to hit is the cell membrane. External to that, if it's present, is the cell wall. And external to that, if it's present, is the glycocalyx. Good job. Okay, so I'm going to give you a second to look at this diagram and think about what all these different parts could be. And then I'm going to cold call people. And how it's going to work is I will call out a letter, let's say A, and then I'll call out a name. And then you'll just unmute and say what you think it is. Or if you're not sure and you don't want to guess, you can say pass. And if you do want to guess, you go ahead and say, I'm guessing it's this. That's also fine. So take a second to look this over in case you get called out on like D or H or whatever it might be. And once you've had a second to look it over, I will cold call some people to answer. Okay. So let's start with A, and I'm going to ask Veronica if you're able to unmute and tell us what you think A is pointing to. And you can always say pass, that's totally legit.
That is A.
This one right here. What do you think?
Is that the one that counts on the DNA?
Yes, that is the DNA. Absolutely right.
Yeah.
So this is the
Yeah.
DNA. It's also called the nucleoid, but that's the region, doesn't have a membrane around it, that has that has the DNA in it. Good job. Okay, I see you, Janelle. Okay, I will cut you some slack with that. I'm so sorry. Janelle can't see the picture. Okay, let's go to Stacy Milton. What would you say is a B? B.
Is it the pilior fimbriae?
Exactly right. So the little projections that poke out of bacteria can be pili or fimbriae. They're made of the same thing. They look very similar. So those are names that are used to describe them. Good job. Okay, let's go to Lily. What would you say is C? What's your best guess as to what C is?
I think it's pointing to the ribosomes.
Yes, exactly right. So there's a ribosome and there's a ribosome, and then there's this little polysome. We'll talk about why those are there, but yes, those are ribosomes. Okay, let's go to Jenna. Jenna, what's D?
That's the flagellum.
That is right. Okay. Dan, I have two Dans, so I'm going to go with Daniel Wandel if you're there. What's E pointing to? I'm unable to hear you. I'm so sorry. I'm going to move on. Edgar, what do you think is E?
Oh, you said E, right?
Mm-hmm.
That'd be the plasma membrane?
You're close. It is the stuff inside of the cell. What would that be if it's inside of the plasma membrane? Oh, I got some hands of people who are desperate to answer. Okay, so I'm going to go to Elizabeth Hernandez. Go ahead.
Is it the cytoplasm?
That's right. Good job, everybody. Okay. What about F, Esmeralda? What do you think F is? F.
Oh, you said F?
F, yes.
Would it be the cell wall?
You're close. It's not the cell wall. Let's see some hands for people who want to. Go ahead, Denise. What do you think?
The cell membrane.
It is the membrane. Good job. Okay. Let's go to Erica Mendez. What's G? If this is the plasma membrane, what's G?
Can you hear me now?
I can hear you.
Okay. Cell wall.
Yes, that's correct. Okay, Haley, what do you think H is?
Can you hear me?
I can hear you.
The name is escaping me, but I think it's the sugary coating on the
It is the sugary coating. Excellent.
Oh, glycocalyx?
Yes, that's right.
Okay.
Fabulous work, guys. All right, I know it's like under pressure. You're like, I'm trying to remember. Okay, so speaking of the glycocalyx, it is a sugary coating. It can be quite thick. It's not found on all bacteria, but when it's there, it's outside of the cell wall. And there are two bacterial types. So just write the names of those two bacterial types in the chat. Good, yes. Capsule is the thick one that's strongly attached, and the slime layer is the thin one that's easy to wash off and kind of loose. Good job. All right, we already talked about this. Not all bacteria have a glycocalyx. Some of them do, some of them don't. But why do we care? What is the clinical or medical relevance? Raise your hand if you want to answer this. Iveline, go ahead.
They help it become invisible from pathogens, so they won't destroy it.
Yes, so if you happen to be a bacterium that's pathogenic, because not all bacteria are, and you happen to have a capsule, it could be one of your virulence factors, and it's helpful because when a bacterium is encapsulated, the white blood cells in your body have a hard time finding it. So Bacillus anthracis, which causes anthrax, Streptococcus pneumoniae that causes bacterial pneumonia, are two examples of human pathogens that are encapsulated, and without their capsule, they become significantly less dangerous. Now, that does not mean that having a capsule means you're a pathogen, because many encapsulated bacteria don't do any harm or can't even live in your body. So it sounds like a few people are having lag issues and some people are not. So it's across the board. If it's really bad, just write that into the chat and we can do something for you. All right, flagella, we're moving on to flagella, just speeding through these cell structures in prokaryotes. What's the function? Write this into the chat of the flagella. Great. Yes, it provides motility in prokaryotic cells. How big is it? What's its width? Not length, that'd be quite long, but what is the width of the flagellum if you know from the video lectures or your readings? I have small, that's legit. 2 micrometers, nice guess, that's way too big, but I appreciate you guessing. Yes, there we go, 15 nanometers, 15 nanometers. Just think about that. That is like half the size of a ribosome or like smaller than a typical virus particle. So it's really, really skinny. Before we move on to composition, let me just ask you, could you see this flagellum with a regular light microscope on doing like a simple stain? Okay, and that's because the light microscope's limit of resolution is what? Write that into chat. What's the limit of resolution of a light microscope? Good, 200 nanometers, 200 nanometers. This is smaller than 200 nanometers, so can't be seen unless you do what? What do you have to do to bacteria to see this under a microscope, a light microscope? Yes, special flagella stain, good job. Now it's made of this protein called flagellin, so hopefully that will be easy to remember. And what I want you to think about it is it's a hollow tube made of this protein. So it's just it's very simple hollow tube made of flagellin protein. How does it move? How would you describe its motion based on your background readings and watching the video lectures? Write that into the chat. How would you describe its motion? Okay, so remember we're in prokaryotes right now. We're talking about prokaryotic flagella only. So the answers that are correct in the chat are rotation. The prokaryotic flagellum has at its attachment point, so there's the cell and it's attached to the cell by a little motor that rotates, and that's how it gets its motion. So it rotates quickly, like 82 miles an hour. We cannot see it with a light microscope unless we do our specials flagella stain, our mascara, like coating with dye and mordant until it gets nice and thick. And the importance, of course, is to the cell is its function, which again, we've talked about several times, is motility, right? So the function of this in terms of the cell's perspective is motility. Why do we clinically care about flagella? Write that into the chat. What is sort of like, what are some of the things that knowing about the flagella is helpful? Okay, so I'm seeing 3 great answers. So identification and diagnosis, and motility in the body, like if it's a pathogen, can it spread? So we'll talk about the identification and diagnostic part. And this is again straight out of your video lecture. The different species that are flagellated have distinctive arrangements. And so, this is Helicobacter pylori has a particular arrangement, and another, it looks like a tuft of hair. A different type of bacteria might have just one on one side. Some of them have one on each end. Then some of them are covered in hair like your hairy uncle, okay? So the arrangement can be helpful. It's not like unambiguous, but it can be helpful sort of confirming our suspicion or ruling things out. And the other thing, and this is a little bit more abstract, right? So we can look at a flagella stain and see this information and be like, okay, that's how many there are and how they're organized. This here is, we would have to have a biochemical information, and it is possible to get this, but basically the flagellin protein itself, so this, remember, it's a hollow tube of flagellin protein. That protein has a 3-dimensional shape that's conferred by the DNA. And you can make subtle modifications to the language of the DNA, to the gene sequence, that alters the amino acid sequence in each of these little proteins so that its surface texture is a little bit different. It doesn't alter its function. It still works as a tube, but its surface texture is different enough that antibodies in the human body can tell the difference. And so we call those differences antigenic differences or antigenic variation. And we can identify which antigenic variant is present on flagella, and that can help us identify who we're dealing with. And a very famous example of this is E. coli 0157H7, okay? Right into the chat if you have like a 1 or 2 word association with this. What does this make you think of? E. coli 0157H7. It's been in the news for the last couple decades, periodically. So someone wrote lettuce, someone wrote Chipotle, someone wrote Jack in the Box, thank you, my generation, what? Okay, meat, sickness, okay, yes. So this is a strain of E. coli that is a little bit different from the strain that's living harmlessly in your gut right now. And this strain has a toxin gene and it makes you very sick. It gives you bloody diarrhea. And it can enter the food stream in a variety of ways, often because crops are watered with water that has sewage in it, or because you eat contaminated meat that's not properly cooked. And we can sometimes tell who we're dealing with because this particular species has antigens on the flagellum. That's the H antigen. And also has antigens elsewhere in the cell wall we'll talk about in a bit. Okay, axial filaments. So if we look at this guy right here, what we see is that the flagellum is internal, right? It's attached at one end. There's a little rotatey thing and it rotates. Because it's inside, it causes this to have a very distinctive movement. And what's the morphology that is caused, that having an internal flagellum called an axial filament, what does that cause the cell's morphology to be? That's right, spirochete, exactly. Okay, and how do spirochetes move? How would you describe the motion of spirochetes? Yes, they move like a corkscrew. Just imagine what would happen if you tie this down, right, inside the cell, and then you start rotating it. The whole thing's going to rotate. The cell is going to rotate like a corkscrew through any liquid. Are there any examples of pathogenic spirochetes or diseases caused by pathogenic spirochetes, if you're not sure of the species name, that you can share with us in the chat? Any diseases that you know are caused by pathogenic spirochetes or the names of those species? Evelyn, go ahead.
I just had a question on the morphology.
Yeah.
I was just wondering, why would you not classify it as a spiralum?
It's a great question. So it's a little bit of a sort of an expert slash subtle distinction, but basically spirochetes are more tightly wound than spiral bacteria. So imagine like just a much tighter coil. And so like on a test, I'm not going to ask you a question that makes you have to dig that deep to figure out, is this a spiral or a spirochete? But really what you need to know is spirochetes are caused, their morphology, their specific motion and morphology is caused by axial filaments. Spiral bacteria have a looser curl and they don't typically have an internal axial filament. Instead, they can have external flagella, if that helps a little bit to understand the distinction.
Yeah, thank you so much.
All right, so this is not something you have to know, but something that's just for fun. Some of you have written syphilis into the chat, and that's accurate. So syphilis is an STD, but it's not just, I mean, it's sexually transmitted, but it's not just, it doesn't just cause like it doesn't just cause symptoms in your sexual organs, and it's not just like annoying. It can be lethal, and for many decades, you know, in hundreds of years in the human population, a severe case of syphilis can progress to something like this, where it invades the craniofacial tissues and your bones, and it erodes your face, and it's just, and then it gets into your brain and causes a kind of really terrible dementia, and you die this agonizing, painful death. Not everybody who gets syphilis, however, dies of it or even has these worse neurological or musculoskeletal symptoms. Some people have just a lesion and then they clear it, and some people are asymptomatic carriers. So it's pretty broad. But the good news about syphilis is that it can be treated with antibiotics. So when that happened, that was amazing. But people suffered for a long time with this disease. It's very contagious and didn't understand how it was being transmitted. And so this is the causative agent, Treponema pallidum, and it's a pathogenic spirochete. This is a skull of somebody who died of this disease, and you can see that it eroded their skull away. Another famous one causes Lyme disease, Borrelia burgdorferi. Again, you don't have to know this for a test, but you've heard of Lyme disease, and so this is, people think, oh, it's caused by ticks, but it's actually caused by the bacterium that is carried by some ticks. Okay, now we talked a little bit about this earlier. So aside from the flagellum, which you can see here, there's other extensions on the surface of some bacteria, which are called what? Write that into the chat for me. That's right. That's correct, fimbriae and pili. Good job. If we were to compare them to flagella, in terms of their size and composition, what would you say? Are they smaller? Are they larger? Is the protein the same? Is it a different protein? Good, yes. So most of you are saying that they're shorter and they're skinnier, which is true. And also they're made of a protein called pillin. So both fimbriae and pili are made of pillin. And they have 3 functions. If you know one or all of them, write that into the chat. What is the function of these extensions? Good, okay. So a lot of you said attachment, that's true. So they can be helpful in grabbing onto tissues. Let's say if you're a pathogenic strain of bacteria and you want to grab onto a tissue like the airway or the urinary tract or something like that, so you don't get washed away by fluids. So attachment. Some of them have an ability to twitch, so they provide twitching motility. It's not as good as flagellated motility, but they provide twitching motility. And some are specially constructed. They're a bit longer and thicker, and they can actually transfer DNA in between cells. So those are the functions of these. Now, if you have a bacterium and its pillin gene gets mutated, or it loses the gene or whatever, then what's the consequence for that bacterium? If you're a pathogen, it means you maybe can't attach as well or can't move around as well. And if you're a member of a biofilm, like not a human pathogen, but just living in the wild, one of the things you want to do in this community is grab onto each other, and maybe you lose the ability to belong to that community. So loss of attachment is the primary issue that will happen. Okay, so we started at we were working our way in, we worked way through the glycocalyx and all these extensions. Now we're at the cell wall. And I think what we're gonna do actually is take our break right now and talk about the cell wall after the break. And we're gonna take just a little bit of we'll take our 10 minutes. So I'll see you guys at 11:03. Okay, so I'll see you at 11:03. All right, welcome back, everybody. Let's cruise right on into the cell wall. So the location of the cell wall is, it's exterior to this plasma membrane, but it's interior to the glycocalyx if the glycocalyx is present. And its main function, as I have asked you many times before, is protection from osmotic lysis. And what's it made of? Write that into the chat. We are in the prokaryotic world. We're in bacteria. What's this cell wall made of? Excellent. Okay. So it's made of peptidoglycan in bacteria. In archaeans, it's made of a different substance. But in the world of bacteria, the whole domain of bacteria, which is many, many, many species, and we care about the composition of the cell wall for a number of reasons, right? One of those reasons is that it can tell us whether or not this organism is sensitive to penicillin. So that's something that you'll remember from both the last live stream that we were in together, but also your video lectures. What is the gram status of the organism type that is sensitive to penicillin? That's right. So gram positives by nature, the particular way that their peptidoglycan is arranged makes them sensitive to penicillin. Is it possible for a gram positive organism to become resistant to penicillin? Yes, absolutely possible. And that's important because what can happen is some bacteria over the course of evolutionary time and evolutionary pressure can develop a protection against penicillin. That basically, they develop an enzyme that chops up the penicillin in a particular part of the penicillin called the beta-lactam ring. And then you now have something that was formerly sensitive to penicillin. Now penicillin no longer works and you need something else. On the other hand, if you are gram negative, you're already naturally resistant to penicillin, and that's because the target of penicillin is just not present in your structure at all. Gram negatives have something in their cell wall called lipid A, and that's important because lipid A is a toxin, and we'll talk more in the pathogenesis section exactly how it works as a toxin. And then attached to lipid A is something called the O polysaccharide. And this O polysaccharide is a long chain sugar. It's a complex carbohydrate, and it can be organized lots of different ways depending on the enzyme that makes it. And so different species of bacteria have different enzymes that make different versions of the O polysaccharide. And those different versions can be detected by your antibodies. And so that's why we call them antigens. The 157th version of the O polysaccharide is found on this pathogenic version of E. coli and helps us to identify it. The 7th version of the flagellin protein is also found there. Okay, so here are the two basic cell wall types in bacteria. There's one that's a thick, multi-layered, cross-linked peptidoglycan containing teichoic acid, and there's one that has a thin, loose peptidoglycan layer that's not cross-linked, and then is surrounded by an additional layer of phospholipids with lipid A and O polysaccharide. So the one on the left is which one? Yes, it's gram positive. And I just want to take a second to acknowledge I saw two comments from students who are unable to hear this. So I'm just going to say, if you can't hear this, you can use the makeup questions and the recording. Email me. Okay. The one on the other side, if this is gram positive, thick, multi-layered cross-linked peptidoglycan with teichoic acid, then this one must be the gram negatives, of course, that has the thin layer, you can see it in brown, single layer, no cross-links, and then an additional phospholipid bilayer on the outside that contains both lipid A and the O polysaccharide. So this one would be what color in a gram stain? What color would the gram positives be in a gram stain? That's right. They would be purple, okay? And that's because when you do the gram stain technique, because of the structure of this cross-linked meshwork of peptidoglycan, the crystal violet gets in, the mordant makes the crystal violet crystals big, the alcohol causes the mesh to dehydrate and condense, and that traps the crystals inside the cell. On the other hand, in a gram stain, gram negatives turn what color? That's right. They turn pink. And that's because crystal violet still gets in, the mordant still causes the crystals to enlarge, but the alcohol washes away this lipid layer, and there's no cross-links, and there's no dehydrating meshwork. And so the crystals just leave again. Now the cells are clear, and then we counterstain them with safranin. Okay, gram positives are penicillin sensitive or resistant? Yes, that's right. Gram-positives are penicillin sensitive normally, unless they have this like protection gene, which would be additional, but normally they're sensitive. And that's because the peptidoglycan meshwork has crosslinks, and the crosslinks are what penicillin attacks. Gram-negatives are naturally penicillin resistant because they don't have these crosslinks. So they don't rely on the cross-linking in order to maintain the integrity of their cell wall. Okay, so I am now finally ready to send you away. I've created your breakout rooms in mostly in to reflect your lab groups so that you can be comfortable and get more time working with these people that you rely on for other things. However, in 1 or 2 cases, the members of the group, there was only 1 or 2 people, so I redistributed you to other groups that were also missing members. So you may or may not recognize the people that I'm gonna send you away. But before I send you away, let me explain what this is going to be about. Okay, so penicillin, we've just talked about how it works. Penicillin works because it damages the crosslinks in the peptidoglycan of gram-positive cells. Okay, now, do all antibiotics target the same thing? Do all antibiotics target the crosslinks in peptidoglycan? That's correct. Not all antibiotics do. So what your job is going to be in your group, you're going to go away and you're going to decide who's doing what things. Okay, so as a group, you're going to pick just one of these. So that means like some groups only have 3 members, some have 4 members. So you won't get to all of these. But in your group, each of you is going to pick one of these antibiotics. So you have 5 options. So hopefully there's no fighting over which one you get. And you're going to decide who's going to do what, and you're going to do very quick Google research to try to figure out what the action of the antibody is. Like, what does it actually do? Which, does it target peptidoglycan? Does it target the cell wall, the cell membrane, the ribosome? Like, what is its target? See what you can find out to the best of your ability. And then you're going to report that back to your group. So you're going to get together, you're going to pick one of these. So take a second right now to just write down the names of these antibiotics. You know which one you want to pick and what your options are. So we've got tetracycline, streptomycin, methicillin, polymyxin, and vancomycin. So those are your options. You're going to pick one of them in your group, and rock, paper, scissors if you need to, if you're both trying to do the same thing. Okay, so I'm going to now open the breakout rooms. Once you've copied these names down and you are ready to talk to your people, just go ahead. You're going to go into your group. You're going to pick one of these. You're going to all decide who does what, just once. So you might only be doing 3 of these, and then report back to your group. And so I'll give you guys about 5 minutes to do this work. And then we're going to come back as a class and talk about it. So you should be receiving your invitations now. All right, we're just closing the breakout rooms, and they'll be completely closed in 20 seconds, and then we'll just set up reconvene and find out what you guys learned in your little research project here. Yeah, I saw that, Christina. I'm so sorry. Oh, Claudia, that sucks. Your computer died. That sucks too. Okay. All right, everybody, welcome back. So what you're going to do now is you had a chance to pick something to research and talk to each other about it. So for some of these, you're not going to have an answer because your group might not have gotten to it. But if you did find out something, as I call each of these, you're going to write in chat what you found out. So what's the action or the target or the mechanism of tetracycline? Just write into the chat briefly what you found. Good. Great. Okay. So a consensus of answers, it inhibits protein synthesis. The target is the 30S subunit of the ribosome. And it prevents translation, which is the same thing as protein synthesis. Good job. Okay, so one of you actually, or maybe more than one, wrote that it is wide spectrum, which is an important thing that we'll talk about in a second. So thank you for also mentioning that. What about streptomycin? What did you find out about streptomycin? Yes, same mechanism, very similar. Also attacks the ribosome. Okay, let's talk about methicillin. What did you find out about methicillin? Yeah, so it has a very similar mechanism to penicillin. It attacks the crosslinks in the cell wall, in the peptidoglycan cell wall. So take a second to think about whether you'd expect that to be wide spectrum, meaning it sort of attacks a lot of bacteria, or more narrow spectrum, meaning it only is going to focus on, say, gram positives or only on gram negatives. Yeah. So it's going to be a narrow spectrum antibiotic by the definition of, does it affect like both gram statuses or just one? What about polymyxin? Did anyone do polymyxin? It's not as, there we go, yeah. So polymyxin targets a different structure altogether. It targets the cell membrane. So that's an interesting one because we also have cell membrane. So we sort of think about, what does that mean? Is that a problem? Or are there differences that are sufficiently specific between prokaryotic and eukaryotic cell membranes that we can actually have an antibiotic that's specific to bacteria? And last but not least, what about vancomycin? Did anyone do vancomycin? And if you did, what did you find? Great. Yes. Again, similar to penicillin. Okay, so what I'm hoping you got out of this was that antibiotics have different targets, and there's a sort of groups of antibiotics that kind of target peptidoglycan crosslinks, and there's some that target the membrane, and some target the ribosomes. And so they're doing different things, which means that they're going to be more useful in some circumstances than others. Great. Okay, so let's have a look at this plasma membrane. Okay, you should be fairly familiar with the structure of the plasma membrane because it's a phospholipid bilayer. It's found in all cells. In the prokaryotes, like all cells, its important job is to regulate exchange, and it does that by providing a barrier to polar things. The phospholipid bilayer is a barrier to polar things, and providing membranes that allow exchange of those things in a regulated way. However, in prokaryotes, as you'll have remembered from the video lectures, there are some additional things that bacterial membranes are often doing that are not done in eukaryotes, because the eukaryotic cell has organelles, which have membranes. And so you can sort of like allocate functions to those membranous compartments that the bacteria would have to do everything at the membrane. So energy production, in particular respiration, some bacteria are capable of doing cellular respiration, and that requires an electron transport chain in a membrane. So they'll put their membrane in the plasma membrane, not in the mitochondrial membrane, because they don't have a mitochondrion. Some bacteria can do photosynthesis, and normally in a eukaryotic cell that's done in the chloroplast, in the membrane of the chloroplast, where you find more electron transport machines. And so those are going to be put into the membrane of a photosynthetic bacterium as well. In cell biology, presumably you talked about how you cross membranes, and so I'm just going to give you a couple of prompts here. So what molecules can cross membranes freely? Instead of doing this as a cold call, I'll just have you write into the chat anything that you know. Something small and hydrophobic, water can cross membranes, gases, ions cannot, right? Then cross membranes freely as in like with no assistance, so ions cannot do that, but gases can, and small nonpolar things can, and things like carbon dioxide and oxygen and water, and also fats, right? Smaller fats, not huge ones, but like, you know, short chain fatty acids, steroids, which are lipid based. Good. Now, what sorts of things would require help to cross the membrane? So they would need a protein transporter or a channel to get through. Yes, things like ions, anything that's polar, things that like glucose and amino acids, right? Now, some of you are thinking, but water's polar, how come it can cross? And that's a whole other conversation we'll just touch on briefly, but is not a main focus of this class. But we will just sort of say, for a long time, it was just known that water can cross membranes, but it was a little bit puzzling because water is polar. And then it turns out that, first of all, water does have the ability to cross membranes, but also often cells have aquaporins as well. Okay, so things like ions, amino acids, and sugars are going to need help crossing membranes. That means they're going to need a protein to do the job. All right, so there's ways for things to cross, like, or circumstances in which they cross, having to do with the concentration, relative concentration across the membrane, and whether a protein is required and whether energy is required. So there's 3 categories of crossing. And so what we call those categories, when something is crossing, it could go by this method, that method, or another method. Good, yes. So simple diffusion, facilitated diffusion, and active transport. These are all ways that you can cross the membrane. So of those 3, what's being depicted here? So here we're seeing things moving from high concentration to low concentration. So they're going down their normal gradient, like they're going in the direction they'd want to go. There's no protein, so they're freely crossing the membrane, which means they must be like water or gases or something small, nonpolar. What do we call this? Yes, that's simple diffusion. So things are moving in the direction that is natural for them. So no energy is required, no protein is required. They're just crossing the membrane freely. So things like water and gases and lipids do this. Energy is not needed because we're not doing anything that is sort of against the natural flow of entropy in the universe. So they're just distributing themselves to eventually reach equilibrium. So there's no energy requirement. Now, in this case, we see that we have things that are still going in the direction that is natural, that is to say from high to low. They're still moving in that direction to kind of reach equilibrium, but they're charged or polar, like sugar or amino acids or something like that. And so they are needing a protein to go through. So what do we call this? Yes, this is facilitated diffusion. And do we need energy? Since it's going from high to low concentration, do we need energy? Does this protein is a helper, does it need energy in addition? It does not, that's correct. Okay, so facilitated diffusion requires protein, but not energy. Okay, so what direction are we moving in this case here? What's shown on the right? Are we going from low to high or high to low? What direction are we going? Yes. So just imagine this is a cell. So here we have this little piece, but here's the rest of the cell, right? And the cell is moving through a world that's relatively barren, right? And it's got resources inside. It would like to keep those resources. And then it runs into like one little glucose molecule and it's like, yoink, I need that. Now, because it's glucose, in this picture at least, and it's polar, it can't cross the membrane freely, which is actually good news because we want to keep these guys inside. So it's going to need a protein friend, okay? But is it going to need energy? Yes, it does. And what is this process called? All right, good job, you guys. You did your homework and you were paying attention in cell biology class. That's excellent. So when we're going from low to high, we really only have the ability to do that for things that are actually unable to cross the membrane freely. If they cross the membrane freely like water or lipids, we can't really keep them in. But if we're going from low to high, we can do that for these polar charged things. And we do need a protein helper and we do need energy because we're going to be bringing things in against their natural concentration gradient. Now, one of the things that's sort of a corollary to that is the movement of water. Water can cross membranes freely. So it moves in the direction that it wants to go, and it wants to go towards the charged polar things. It's attracted to those things. So if we look in this situation right here, where water is moving in and out of the cell pretty equally, and the reason for that is that the outside world and the inside world have roughly equivalent amounts of sugary polar, charged things that the water likes. So this situation is called what? That's right, good job. This is called isotonic, when the concentration of the stuff water likes in the inside of the cell and the outside world is the same or similar, and water is just kind of flowing in and out with no particular direction. This is isotonic. Now, this situation is one where you can see and this is very common. This is extremely common. The cell has a lot of goodies that water likes, like sugars and amino acids and ions. And the outside world is less so. It is more watery, it has less stuff, okay? And the cell is, obviously, water is attracted to the inside of the cell. So water is trying to move inside overall. That's its overall kind of net movement. So what do we call this type of situation? Correct, yes. This is hypotonic. When the world is watery, it's hypotonic. And this is the dangerous time because if water moves in and the cell has no protection, it's going to explode. Now, what about over here? We also see another situation that's a problem. The outside world now is very salty or sugary or something is out there. And we have all this water is leaving, we're becoming dehydrated. This is known as being what? That's right, that's hypertonic, when the world is salty or sugary. So what happens then is the cell is dehydrating, and that dehydration can be lethal, which is why things like Twinkies can last forever, and sugary, salty things are well preserved compared to other things. So what is it in bacteria that prevents bacteria, which are in this sort of often in a hypotonic situation, not always, but often. So why don't they explode all the time? What's protecting them? That's right, it's the cell wall. And so this is one of the reasons that some antibiotics are useful. They target the cell wall, they make it more possible for this bacteria to explode in these very common situations where it's in a hypotonic environment. Does the cell wall protect bacteria against this hypertonic situation? Does the cell wall prevent the motion of water out of the cell? That's right, it does not. And that explains why pickling things or packing pork with salt for a long ocean voyage before refrigeration, electricity, and gas, and why baked things that have a lot of sugar in them can be left out at room temperature longer than, say, like a pork roast, that has relatively little. So this is why salting or sugaring things is preservative, because it dehydrates a lot of the bacteria and they are unable to protect themselves from that. Okay, last little thing we'll see. So let's take a freshwater protozoan, amoeba, okay? Freshwater, meaning it lives in something like a pond. So it's happily in its environment. What would this be considered? Great, yes, that's an example of isotonic. Let's take E. coli, and it's found on the surface of, say, a pork chop, and we pack that with salt and put it in a barrel. What kind of condition is this E. coli in now? Great. Yes, it's hypertonic. It's going to dehydrate the E. coli. Take human red blood cells, okay? We're in the bloodstream now, so just put yourself in that floaty bloodstream, and you're a human red blood cell, and then you're in an OR, and they inject you with sterile water instead of saline by accident, which has happened because those bags look so similar. And now what's going to happen to your red blood cells is they're going to explode because you are hypotonic. That's right. It's very dangerous. Don't give your patients water instead of saline. Bad idea. Okay. Now, if we look inside of a cell, there are some other interesting things that we have to pay attention to, such as the fluid-filled interior called the cytoplasm. That's all this stuff. The DNA, which is, what's this area called? It's not surrounded in a bacterium by a membrane. What do we call this area where the DNA is found? That's right, it's the nucleoid. Okay, so I am sorry, right now I'm just gonna say this many times, I'll say this, but There's a lot of very similar words in biology, and it's very annoying. Okay, so this is not the nucleus, this is the nucleoid, and it's not the nucleotide, and it's not anything else that sounds like that. It is the nucleoid, that's its name. And then these little dots are ribosomes, which do the job of protein synthesis, right? Okay, so we have to talk about the ribosomes for just a minute, and hopefully your little breakout helped you understand them a little bit better too. We can see they have two pieces, a small piece and a big piece, and they have these S coefficients that refer to their density. So you can measure their density. This has a density of 30 and this has a density of 50. If you stick them together, what density does this whole particle have? Write that into the chat. Yes. Prokaryotic ribosomes have a density of 70S, which is annoying because it does not add up, right? 50 plus 30 is not 70. And that's just because density is not linear. When you stick a 30S and a 50S thing together in one unit, they become a different density than that you would imagine that they would be. So the prokaryotic ribosome has the job of doing your protein synthesis. It's made of RNA and protein chunks, which you can't really see here, right? So, but just imagine this has a whole bunch of building blocks and they're all different pieces of RNA and protein. And it's got this density of 70S. It's got these two subunits, 50 and 30. And yes, you do have to know this. And part of that is because is this information that I've just given you the same as a eukaryotic ribosome? Are there any things about this that are the same? Anything about what's written here that's the same for eukaryotes? Function, protein synthesis, correct. What about this? Is a eukaryotic ribosome also made of RNA and protein? Yes, it is. Okay, so its function and composition is the same, but the size or density, you know, size is just easier for us to understand, even though it's really density. The density is different. Eukaryotic ribosomes have two subunits, 40 and 60S, so they're denser. And when you stick them together, it makes 80, which is extremely annoying because 30 and 50 makes 80, and this would be 100, but it's just the way it is. So eukaryotes have 80S ribosomes. We'll talk more about that on Thursday. Another thing you might find inside of a bacterium is an endospore. You can see that picture here is a nice rod-shaped guy. You can't see it's in a chain. So here's the cell wall, and you can't see the rest of the cell wall here. This is a nice rod. It's connected to another rod, and this one has an endospore. So write into the chat the function of the spore for me. What's the function of the endospore? Yes. So it provides protection from adverse conditions. And here is how it works. So just in case this wasn't clear from the video lectures, when a cell is going to make an endospore, what it does is it goes through sort of an aborted, modified version of binary fission, which doesn't involve fission. So it goes through the first part, copies its DNA. And then around one of those copies of DNA, it just puts a very tough spore coat. And it's sort of basically saving, it's like archiving its DNA for if shit goes sideways, bad things happen. And so if that were to happen, you know, and the cell can't survive dehydration or no food or UV radiation or antibiotic exposure or whatever it might be, the cell dies. The endospore is so well protected and it's not metabolically active, so it doesn't need any food or water. Then its DNA gets preserved until conditions improve, and then that little spore can germinate into a regular cell. So it's this like tiny mini me version of the cell surrounded by a tough spore coat that's protecting the DNA from all of these potentially lethal conditions. In the chat, just write in the names of the genuses from the video lectures, the two genuses that are common that contain species that make spores. Endospores. Yes. Yes, the two genuses are Bacillus and Clostridium. So within the Bacillus genus, things like Bacillus anthracis, but a bunch of other Bacillus species, right? There's tons of them, many, like 200, can make spores. And within the Clostridium genus, a lot of those species, Clostridium tetani, Clostridium botulinum, and many other Clostridiums can make Clostridium diphtheria. They can all make spores. So you don't have to know the species, but you should know these genuses. Now you don't have to know this, that species that are human pathogens. I'm telling you this part for fun, because woo, the fun. Bacillus anthracis is a famous human pathogen that's in the Bacillus genus. There's lots of bacilli that live in the dirt and would never harm you. Clostridium tetani, causing tetanus. Clostridium botulinum, causing botulism. Clostridium difficile, or C. diff. Many of you are familiar with that from healthcare. Okay, so we already did our little antibiotics lookup and kind of talked about narrow and broad spectrum. So I won't belabor that. I'll just sort of very quickly say that when you're looking at the mechanism of action of these antibiotics, which you will do again in week 5 or 6, I think, you'll sort of reconsider this. You can sometimes guess if they're going to be narrow or broad spectrum, or understand why they're narrow or broad spectrum, because, you know, for instance, some of these only target structures in gram positives, and they would be narrow spectrum because they're not going to get all of the bacteria. And others target structures that are shared between the 2 gram statuses. Now, these terms actually have more specific meanings, and I don't want to burden you with that, but we'll just use a very simplistic definition for this class. So just off the top of your head, which of these do you think, just write one into the chat, that you think would be narrow spectrum based on what we've talked about? Yes. Vancomycin and methicillin are 2 examples of ones that have a very similar mechanism to penicillin, and they target gram positives. And they've been deployed sort of successively because the initial penicillin sensitive gram positives, some of those species developed resistance. So then we sent this guy after them. And then they developed resistance to this guy. And so we sent this guy after them. And now we're in a position where some of them are immune to, or resistant to that. And some of these other ones you might predict would be broader spectrum because their target is, say, protein synthesis or the membrane, which is something that is less gram specific. Okay, I am actually going to check our time here. I think we're going to save the Kahoot for next time because we're going to run out of time otherwise. I want to talk about the unknown ID project in the last few minutes of class. So we're finishing lab 2 this week, and I talked about that last week. What we're starting this week is the unknown ID project. And the very first thing that you need to do is claim your ID number. That's something you need to do by this weekend. I would do it sooner. I'll show you exactly where to find it in a second. But once you've done that, you want to read the background and the part 1 and part 2 instructions for this project. There's actually 4 parts. So you're going to read the background. That's going to talk to you about making a lab notebook that you'll keep. You're going to read part 1, and what you'll do for part 1 is, after you've claimed your number, you'll ask the technician to send you a picture of your gram stain, and you'll figure out what that is. You'll use that actually to determine your shape and gram status, and then pick a dichotomous key, which is described in part 2, and you'll use your dichotomous key to find out what your group is. So this part will take you longer than a day, okay, and maybe a little bit more complicated. And so we can talk more about this on Thursday. But for right now, claiming your number, which I'll show you how to do right now, starting a lab notebook and requesting your gram stain, that's sort of like what I would encourage you to do in the next 48 hours. And then on Thursday, we can talk about this thing. How do we actually go from the gram stain and move forward from there? Once you find your group, you're going to be in a group which has somewhere between 12 and 200 options. And so you will do additional work to figure out what your exact species is. At the end of that, you'll have a guess, an educated guess, and then you're going to ask for some sequence data and see if it supports your guess. Okay, so next time we're going to talk about eukaryotic organelles and structures. Right now what I want to do is just take the last few minutes and show you exactly where to go in Canvas to get started on this unknown ID project. So I need to do a new share. Okay, so here we are in our Canvas course shell, you're going to go to modules and you're going to go to week 3. And what you'll see there is in the lab section, like information that you should read. And then this sample sign up. So let's open that up and have a peek. So what we can see is it's a wiki. You can actually go press the edit button and put yourself into one of these slots, okay? And so then you'll have claimed a number. Do please try very hard not to write on top of each other. And so every once in a while you want to just double check that this is still preserved correctly. There is a way for me to go back and look at all the history of this page and figure out who actually claimed a number first. I don't want to have to do that unless I have to. So just be very careful. Select a number and then make sure that you haven't written on top of anyone else by accident. Save your changes and then exit out. Okay, so once you have your number, what you can do is, if you go back in the background, or in the next page, I'm sorry, in part 1. Once you have your number, you're going to read these instructions and it's going to say, start a lab notebook and then request a gram stain. So your lab notebook, you want it to be a digital file that you can keep. Word, Google Doc, you know, if you want to write it down, that's also okay. But you will then have to scan it into a digital format when you submit. So it's better if you can do it in Word or Google Doc. Something that can be submitted as a PDF ideally, or as a Word document at the end of the quarter. And what you're going to start is every time you do work in this project, every single time that you order a test or receive data or interpret data or think about your project, you're going to have a dated line item in your lab notebook that says date and what you did. Okay, so this is going to be what your first thing will look like. Google Docs is fine, Kelsey, yes. So you're going to have like, you know, October 3rd, 2020, selected unknown ID sample number 4. That's your first line item in your lab notebook. And then you're going to go email to our technician 2, who is doing a phenomenal job of this digital version of an unknown ID project. We just owe her a huge debt of gratitude for this. You're going to email her and say, hi, this is Heidi. I have sample number 4. Please send me the gram stain. And she'll send you a picture. And then you're going to put your picture into your lab notebook. So you go October 4th, 2020, received the gram stain, and here's the picture. You'll put it into your notebook. And then you're going to write what you think it looks like. What is this gram status and what is the morphology? And then you'll read through the rest of this to figure out what to do next. Okay, so basically you're going to use the information as to morphology and gram status to decide which of these dichotomous charts to follow. And we'll talk more about this on Thursday. Okay, so in the last few seconds, I just want to open this up for questions. If you have questions about this or anything else, or our test that's coming up next week, or whatever's on your mind, then I'm here to answer your questions. Otherwise, please sign out, and thank you so much for coming today. If you had technical difficulties and you will need to email me to talk about that, then please do that. And let's start with Claudia. Your hand is up. Go ahead.
Is there any specific way you want us to, sorry about that, you want us to set up our notebooks, like table of contents, stuff like that?
Oh, it's a good question. All I need as a bare minimum is every time you do anything in this project, whether it's requesting a test, whether it's receiving data, whether it's choosing a dichotomous chart, whether it's writing notes about your interpretations, that you have a line item, at least one line item that's dated that says on this day, this is what happened in my project. And other than that, really, it's up to you how you want to organize it. Just it has to be complete. And some people write a lot, you know, and some people are very succinct, and that's fine. If you want to do it in a table format, that's also okay. But it does have to be complete. So it should have everything in there, if that makes sense. So I can follow your logic and I know exactly what data you received and when things happened. Does that help?
Yeah, that's helpful. Thank you.
Awesome. Kaylee, you had a question. Go ahead.
Hello. Can you hear me?
I can hear you.
Okay, cool. I've been having a lot of problems with Canvas for some reason. So it's okay on my computer, but I do a lot of work on my iPad as well, and it won't let me open our
I know.
our section.
I'm so sorry. So, you know, from the beginning, Canvas has never been 100% compatible with mobile devices, never. And that's why I always tell people, do not take your test on a mobile device, because it's done weird things in the middle of someone's exam. It sucks. But this quarter, for some reason, I think there's an update, and not all devices are compatible with the update. So people are having trouble accessing modules and all kinds of things they normally would able to do. And it's not anything that I've changed, and it's not anything I can fix right now, although I'm talking to the IT people and seeing what they can do. But it really is, unfortunately for right now, means that a lot of the work you'll need to do on a computer, which may not be ideal for you. You can get a Chromebook from the campus if that's just a really, like, you need to be more mobile and it's just really a hardship to be stuck to a computer, which I would understand. So I will keep you posted, but I am really sorry. I don't have an easy fix for that.
Oh, that's perfectly fine. I just do a lot of my written work on my iPad, so yeah.
I know, and it's so convenient. I'm so sorry. I know I've heard from several people who are kind of frustrated, and I feel your pain. It sucks. So we'll keep working on it, and hopefully they'll come up with another update that will fix the problems, but I don't know. I'm curious, Kaylee, though, are you in any other classes that use Canvas, and are you having problems in those classes too, or is it just in my class?
I'm also in the HIV/AIDS course, and I haven't really seen any other problems, but I can double check it today because it's only within like the last couple days it's decided to not open the modules.
Yeah, and would you do me a favor, and when you do that little test, can you just tell me what your result was? Like, you know, your little scientific experiment. I would really appreciate that, because if there is anything that I can do in my course, and I can contact another instructor and say, hey, what did you do different than me? There might be a workaround that I'm not aware of. So just keep me posted.
I actually just did it right now, and it's letting me open some things like quizzes for that class, but it won't let me open like lecture pages.
Yeah, what a bummer.
So yeah, I don't know.
I am
I
so
don't mean
sorry.
to interrupt, but I think we got an email or something that posted that they were having troubles with Canvas on like mobile devices, like your phones and stuff, because it was working fine and then it stopped working. So I think it's actually a system technical issue, not just your class. If I'm
okay. Well, I appreciate that feedback. Definitely, guys, keep me posted as to what your experience is, and I'll keep working with the Canvas IT people and see what we can do. But you know, your best bet is always to go to a computer when you're in the middle of something and it's like not working. So yeah, man. Hopefully things will improve. It's never ideal, is it? It'd be better if we can just be in a classroom together, but it is what it is. Thank you for asking that, Kaylee. I appreciate it. And definitely keep me posted what's going on for you, good or bad. I'm curious to know. Okay, Cambria, what was your question?
Are you considering holding a review for the exam?
Let's see. I may. I don't know off the top of my head what might happen. It seems like I'm just sort of thinking about, I try to hold office hours at convenient times, especially ones that are convenient for the online only section. And so it's likely that I'll hold a session that I'm sure most of them can attend, and you guys would be welcome to come to. So that's the most likely thing that I'll do. And if it is working for them, it's probably going to be, if I think about my survey for them, like Thursday night or something like that. Also, I'm happy to set up, if you guys need my intervention, that you might not, Zoom sessions for people to go to even though I'm not around, to talk and study, if that's helpful. So that's a great question, and I will keep you guys posted for sure if anything formal gets scheduled.
Sounds good. Thank you.
You're welcome. Iveline, what's your question?
I just had a quick question on calculating the cell sizes for our lab 2.
Yeah.
I was looking at the images, and on top of the images, it says that the image is like blank micrometers wide. And I was just wondering, do we have to take that into consideration for a calculation, or do we just do the field of view for 100x objective divided by how many cells fit across?
Yeah, great question. So yes, the answer is, if you were doing this calculation looking through a microscope yourself, like in a physical lab looking through the microscope with your eyeballs, you would use the field of view calculation. or if you were doing it from the simulation, right? Where the simulation simulates looking through the barrel of a microscope. For the digital part that I have put together for you, those 6 images, you need to use the information on the, that says this picture is X whatever micrometers wide, because it's a smaller section of what you would see with the, you know, with the oculars. So you can't rely on this picture is as wide as the field of view. It's probably not. Does that make sense?
So then would we treat that like that number as our field of view and just
Yes.
take that divided by the number of
Yes.
cells?
Yes.
Okay.
So, or another way to say it is, you treat that like your scale bar, but your scale bar is the width of the picture.
Okay.
Does that make sense?
So I guess I kind of got confused now. If we treat it as a scale bar, would we have to do the calculation you showed us in the second video of the number of cells that fit across the scale bar, or can we just like count?
So it's basically, it ends up being the same math. So it's just a semantic thing that I'm sorry, I didn't mean to confuse you. If the image says, let's say the image says it's 70 micrometers wide, right? Then you just know that the distance from side to side is 70.
Okay.
Right? And so then you're going to say to yourself, well, how many cells would fit across this whole image? And then you're going to take 70 divided by that number, and that gives you the measurement. Does that help?
Yeah, that helps a lot. Thank you.
Yeah. And if you have trouble, you know, there's a couple of ways to get more precise about it if you're feeling like kind of frustrated by that, trying to do it like just in your head. But it involves importing the image into like a graphics file, a graphics program, and making a little like, you know, little line that's the same size as the cells, and then stacking those lines across your image and then counting them. But you don't have to do that. You can get a pretty good answer with just trying to guess how many would fit across.
Cool, thank you.
You're welcome. Okay, April, what's on your mind?
Hi, can you hear me?
I can hear you.
Is it possible, kind of piggybacking on not being able to view things on our mobile devices, is there any way you could put some of the lectures that are in the modules in the Pano area? Because I can view that, so I can rewatch the lectures or the Zoom calls. I can rewatch those, but I can't watch the original lectures.
I can't easily do that without a ton of work on my part,
Okay.
but let me think about what other things I could do instead. So would you shoot me an email and I'll play around with a couple of things today. I can think of some things that might work, and then I'll just see if they do, and then we can go from there. So shoot me an email, remind me that we had this conversation, and just say, hey, here's my specific issue, what I'm not able to see, and are there any other ways to display this? I can't guarantee that it's yes, because if it turns out that the answer is I need to do something that's going to take me hours and hours, I won't be able to do that. But if there's something simple I can do, like some of my stuff is on YouTube, and so I might be able to send you a link to get you hooked up with that, and it might be an easier way for you because everybody can get to YouTube.
Right.
So let me shoot me an email to remind me, and I'll look at that later today and see what I can come up with.
Okay, and I had one more question. Is the test open book?
Yes, so great question. And we'll talk about this on Thursday as well, but basically the test is open book, right? As long as you're not connected to the internet, which is to say, of course, you're going to be connected to the internet because you're taking the test on Canvas, but you're not allowed to Google things, and you're not allowed to call people on the phone or use your mobile device to Google things. You are allowed to have your textbook next to you, or have a device that's not connected to the internet that just has static notes on it that you've taken during the course of the quarter or something like that.
Okay, and following, because I noticed there's a study guide, so following that study guide and actively studying that study guide will be the most helpful thing, I guess?
Yes, and for the things, so there's like, you know, a variety of different types of ways you'll be tested. Like there's some sort of basic recall things, like do you remember what this is, you know, this structure, what its function is, or do you know what peptidoglycan is made of, or whatever. But there's also going to be some more like synthetic questions where you have to put different types of information together and use logic to solve them. And then there's going to be some questions that are math-based. And for those ones, I've tried to create a couple of examples at the end of the study guide, so you kind of know in general what those questions might look like. Like not every single one of them, but what would a question of that type kind of look like, so you're sort of prepared mentally for that type of question as well.
Okay, thank you very much.
You're welcome. Kylie, what's on your mind?
So I have a question about the lab kits. I can't remember if you said it was gonna be in there or not, but like a mini microscope, if that was supposed to be in there now.
Oh no, so we had for the last 2 quarters been using those, and I decided to dispense with them because they're actually one of the more expensive components in order to allow us to do some other things later in the quarter with plates, because the plates are also expensive. So I decided to get rid of the microscope and instead use the plates. So you do not need a microscope in your kit. Have you received your kit?
Yeah, I received a kit. I was just confused.
And so you're just kind of like, where's my microscope? If you do want one of those little scopes, the part that was expensive was the prepared slides, but the scope itself you can get for $2. The other reason we decided against it is that many places around the world are using this now. And so it was so backlogged, we wouldn't receive it till November.
Yeah.
Like way too late. But if you want one, you can order it from the company. And if you're super interested, you just shoot me an email, I'll send you the information. I also have an extra one for anyone who's like dying to try it out. But yeah, you don't need one for this class.
Okay.
All right, guys, I don't see any more hands up, but if you are not able to press the hands up button and you do have a question, then go ahead and just unmute and ask me whatever's on your mind. And otherwise, go ahead and sign out. And thanks for joining us today. It's been amazing, and we will see you again on Thursday.